We are walking atop a living, breathing, sentient, subterranean architecture that processes information faster than any supercomputer. By mapping how these fungal threads trade nutrients for data, we reveal a biological internet predating humanity by eons. We must decode this silent neural network, proving that the forests think, feel, and breathe as one. The claim sounds impossible, yet the intuition behind it is real. Beneath every step lies a network so dense, adaptive, and responsive that it blurs the line between organism and system. The ground is not a boundary, it is a veil, and just below it unfolds a structure that has been shaping ecosystems long before humans walked the land. To understand what is actually there, the language needs to shift from metaphor to mechanism. Mycelium is the living body of fungi, composed of microscopic filaments that extend through soil, wood, and organic matter, punching at their tips and fusing with one another to form networks that can span entire landscapes. These threads are thinner than a human hair, yet in aggregate they form a mesh so extensive that a handful of forest soil can contain kilometers of growth, each segment alive, each tip sensing nutrients, moisture, and chemical signals. The visible mushroom, the part that pushes into the air after rain, is only a temporary structure, a reproductive organ that disperses spores before dissolving back into the system. The organism itself remains below, persistent and continuously active. When viewed under magnification, the network does not resemble roots so much as circuitry, because the way it grows, branching, connecting, rerouting, produces patterns that echo systems we recognize as information processing. The comparison to a neural network becomes obvious when observing how signals move through these filaments. They conduct tiny electrical impulses, changes in voltage, that travel along their lengths in response to disturbance, resource availability, or interaction with other organisms. These pulses are not random. They encode differences, propagate change, and influence behavior across distances that would otherwise isolate organisms from one another. Alongside electrical signaling, chemical exchange flows through the network. Carbon, nitrogen, phosphorus, and a range of signaling compounds move along concentration gradients, redirected at junctions where conditions shift. A tree rich in photosynthesis output can pass carbon through fungal partners to a shaded neighbor. A plant under attack can release compounds that alter the chemistry of nearby tissues, priming defenses before damage spreads. Long before microscopes revealed the filaments or experiments traced nutrient flows, many indigenous and traditional knowledge systems described the land in ways that now sound strikingly aligned with these discoveries, not because they identified fungal networks, but because they recognized patterns of interdependence that modern science is still learning to quantify. Forests were understood not as collections of isolated trees, but as communities in which relationships determined survival. Around 450 million years ago, when the first plants began colonizing terrestrial environments, the conditions were hostile. Thin soils, limited nutrients, no established cycles to support growth. Fungi formed partnerships with these early plants, extending their reach into the substrate through filaments, extracting minerals, and trading them for sugars produced by photosynthesis. This symbiosis was not optional, it was foundational, enabling plants to survive and spread, gradually transforming barren landscapes into ecosystems. Over millions of years, these relationships diversified and intensified, giving rise to the vast networks that now underpin most terrestrial life. What began as a microscopic exchange became a planetary infrastructure, one that continues to mediate the flow of energy and matter across ecosystems, linking organisms in ways that challenge the notion of individuality. Technically, the system operates through a combination of physical growth and chemical exchange. Filaments grow at their tips, guided by environmental cues, branching when resources are abundant and retracting when conditions deteriorate, constantly optimizing their structure for exploration and efficiency. At points of contact with plant roots, specialized interfaces form, allowing the exchange of nutrients. Fungi deliver minerals and water, plants provide carbohydrates. Within the network, resources move along gradients, but the pathways are not fixed. They are adjusted dynamically as conditions change. Electrical signals, tiny fluctuations in voltage, can propagate along filaments, triggered by mechanical damage, changes in light, or chemical stimuli, altering growth patterns and resource allocation. The temptation to call this intelligence is strong, and it is justified. Mycelial networks integrate signals, allocate resources, and modify their structure in response to changing conditions, achieving goals such as efficient foraging and resilience without any central decision-maker. They can remember in the sense that past interactions influence future responses, with previously explored pathways being reused or avoided based on outcomes. Experiments have shown that fungal networks can navigate mazes, optimize routes between nutrient sources, and adjust their growth in ways that maximize efficiency, behaviors that, when observed in other contexts, would be described as problem-solving. Yet the mechanism remains entirely biological, grounded in chemistry and physics. As research continues, the details of these interactions are being refined, revealing both the power and the limits of the network. Not all exchanges are cooperative. Fungi can also act parasitically, extracting resources without reciprocation, and plants can favor certain partners over others, shaping the network according to their own needs. The system is not uniformly benevolent. It is competitive, selective, and context-dependent, reflecting the broader dynamics of ecosystems where cooperation and conflict coexist. Some species can survive extreme conditions, including high radiation, desiccation, and temperature fluctuations, and fungal spores have been shown to endure exposure to the vacuum and radiation of space in experimental settings. This resilience raises the possibility that fungal-like organisms could persist in environments where more complex life forms cannot, making them candidates for early colonizers in extraterrestrial ecosystems or for use in human-led efforts to sustain life beyond Earth. The idea that networks similar to mycelium could exist elsewhere is speculative, yet it is grounded in the recognition that decentralized, resource-efficient systems have advantages in harsh conditions, processing information through local interactions rather than relying on centralized control. As the concept of mycelial networks enters broader awareness, it often becomes a canvas for projecting ideas about consciousness, intelligence, and connection, sometimes stretching beyond what evidence supports. The truth about mycelium is not that it is a hidden brain beneath our feet, but that it reveals a different model of life, one in which connection is as fundamental as form, and where the boundaries we draw between organisms are less absolute than they seem. I post videos that break down how reality actually works from your brain to your body to the world around you, so if that sounds interesting, subscribe and I'll see you in the next one.